Thank you so much, Mrs. Tucker. Thank you so much for the opportunity to come here today to remember what it is to be a dairy scientist. And my talk today will focus on challenges and opportunities in dairy genomics. I'm scared of the geneticists here because they're really good. You are very well known around the world, and it is an honor, truly an honor to be here today. So thank you, Barry, for the invitation. I watched the lecture from last year, and I know Dr. Rupert Brookmeyer quite well because I actually also was a tenured sort of lecturer at the University of Bern up until me trying to quit 2 years ago and me finally quitting there last year. So Rupert's talk was very good, and I'll try and hit the mark today. This is the formula that I spoke about this morning with your graduate students. We have this I'm not really good at math, so we try to keep it very simple. And this is kind of the formula that we use to think about genetics and maybe nutrition and other areas of dairy science. The phenotype is equal to the genotype plus the environment. And things like milk production are equal to the pedigree plus maybe the environment that the animals are in. In today's world, we've done a really good job of adding more information. Through technology, we can say, okay, our phenotype is not just the amount of milk, but it's actually the contents of the milk, the fatty acid chains, the types of fatty acids that we were talking about this morning. The genotype is not only the pedigree of the animal, but we can get right down to the DNA level. We can actually get down to the bonds of those nucleotides. And the environment, of course, is no longer simply a person hand milking a cow. We can get down to new types of measurements, et cetera, et cetera. And this is really exciting. For the graduate students in the room, you guys are going to be fantastic. You have all of these tools at your disposal, and it's going to be great. But use these tools wisely is my first thing. We have chosen animals based on selection goals. The fundamental part of being, well, there's one more thing that I have to say back here, and that is that nutritionists and behavioralists and geneticists will fight about which component of this is more important, the big E or the big G. The truth is, they're all important. It's all important. If you have a trait with a heritability of something like 30%, it means indeed that 70% of the variation is coming from somewhere else, meaning that environment is truly important. And geneticists have been unable to admit this for many years. I'll admit it. I'll admit it. Sorry, Rob. They're all important. But the question is, how do we partition this as best as we can? So how do we figure out what part is truly genetically based and what part is based through the environment, and how do we control those different things? So in the past, up until relatively recently, maybe that's even within most of our lifetimes, we were focused exclusively on production and conformation. How do animals look and how much milk do they produce? Those were really important things because that's what you get paid for directly. It's a very clear linear relationship. However, as technologies evolved and changes came about, we could start to add things like fertility traits, health traits, workability traits, things like milkability, et cetera, et cetera. And this graph is kind of a weird thing, but it shows the sort of the percentage of the breeding goal that's filled up by these different things. So here this is an older graph from 2017, but you can see that the breeding goal has now incorporated all of these extra things that wasn't the case before. And today, we can actually take all of the different traits that are being measured. This is a Canadian example, but all of the traits that are currently in our index, and we can look at the relationship between those individual traits. So when someone says to you, when Rupert, for example, who I've had many fights with about this, he says, the geneticists are breeding for the energy to go into the udder. And that's why our cows are not fertile anymore. Is that true? I would love to hear. How many people think that's true? Nobody is willing to put up their hand. or they all have shoulders that are sore because they threw Baes. I don't know. So nobody believes that. I don't know if it's true. Maybe. Who knows? But we can start to quantify the relationships between these different traits, which is really interesting because we can measure these traits, we can quantify the relationship between them, and we can come up with a better, more balanced breeding goal overall. Fancy stuff. At the end of the day, however, our producers still need to make money. We have to have a profitable industry in order to move forward. And we can measure genetic gain per year with this formula, and we have different components here. So genetic variation is how much of the variation is caused by the genes of an animal. Then we have selection intensity. That has a lot to do with how much risk you're willing to take. If you just want to select very few animals, you're leaving out a whole bunch of other animals. There's a risk associated with that, but you can make pretty quick progress if you're doing that. Accuracy, this is along the terms of garbage in and garbage out. If you're measuring things that are not real or you're not measuring them well, you're not going to get a good breeding value. It's pretty clear, right? And then, of course, how long does it take you? Generation interval in years. How long does it take you to replace the generation that you're using with the next one? And genomic selection, who's heard of genomic selection? Okay, now I know your shoulders aren't broken. You have heard of it. That's good. Everyone should have heard of it. It's using the information in the DNA to select the next generation of animals. Genomic selection has revolutionized livestock breeding. Truly, because of these reasons. Genomic selection can't alter genetic variation, per se, but it can significantly increase selection intensity. It can significantly improve the accuracy of selection, and you can significantly reduce the generation interval. Now, this bullet point here, genetic variation is not altered. That's true. But there are different ways of thinking about specific traits. And this is also where quantitative geneticists in the past have maybe failed because we're not really the most social bunch. We don't talk to veterinarians. We don't talk to nutritionists. We don't talk to animal behavior people. We kind of stick to ourselves a little bit. Had we spoken with these people a little earlier, we could have better identified traits that we can use, or phenotypes that we could use in order to improve important traits. So unless a more accurate phenotype is developed, genetic variation cannot be altered. But if we're measuring the wrong thing, then it's pretty tough to grasp what it is that you're trying to change. So just to prove, again, that genomic selection works, we can see here on this graph the genetic gains in reliability that have been achieved through the implementation of genomic selection. And I don't know if you can read all these, but what I can tell you is that those are different traits in the breeding value index for dairy cattle, all the way from milk, rump, herd life, somatic cell score, where Dr. Tucker was probably part of instigating that whole phenotype as a trait available for genetic selection, all the way through to metabolic disease resistance. The light blue values here show the reliability just achievable through parent average, so using that pedigree information. And then if you add genomic information, you can see that reliabilities improve between 30 to 40%. That's huge. That's huge. Across the board. Now, when we think about traits that are difficult, those traits like health traits or fertility traits, those are the ones that are still, the gains are clear, but they're lower than for the easier traits, less complex traits. So the focus today, I told Barry I would talk about challenges. So the focus of my talk today is how can we improve these multifactorial, low heritability, and hard to measure traits? What can we do about them to move our industry forward? And I've chosen 3 examples, and I'll walk through those with you today. So are there any reproductive physiologists here? Uh-oh, 1, 2. Okay. So what are reproductive traits to a geneticist? That's all the stars, right? So here we have reproductive traits in Canada. Okay, we have a heifer here, and we have one measurement, and that is age at first service. That's what we use as a trait. How old is this animal when we put semen into her the first time? We might have another, the AI stands for artificial insemination, not avian influenza or artificial intelligence, okay? Just to be clear. So day 56 is a special day. That's when we measure a second trait called non-return rate for heifers. If the animal is pregnant, it's okay. If she's not pregnant, it's not okay. So that's just a 0 or 1 binary trait. Then we have first service to conception. Fantastic, she's pregnant. So this is a date from the first time she's inseminated to the day she gets inseminated. Surprise, there's a calf that's born. Yay. Then we have an animal that turns into sort of a cow, right? Because she's not a heifer anymore. So we have, again, calving to first service, another insemination, day 56 again, but as a cow this time. And then we have first service to conception, and we start the process again. Who thinks these are great traits? This is it. This is reproduction to a geneticist. Who thinks these are great traits? Arms are broken? No. You say yes? Great. Anybody else say yes? Great. Can we do better? Everyone is nodding. Yes, we can. And that is exactly where the challenge meets the opportunity. So can we do better? First of all, we can look at repro traits, right? Geneticists love to show trends. Okay, so we've got this trend. Days open, the trend in the last maybe 10, 15 years has been a reduction in days open of 10 days. Great. We look at the other traits. Okay, calving to first service, the reduction is also 10 days. That's kind of because those two are together with each other, right? We have calving interval here. This is numbers on the secondary y-axis. Also pretty much 10 days. But then looking at first service to conception, we've only been able to reduce that by 1 day. Is that good? Are we happy with that? Can we do better? Maybe. Well, let me think about what this could be. Okay, why could this be? The first thing is, well, I think we can do better personally. And then my first question is, are these trait definitions pretty far away from the biology of the cow? I think they are. I'm sorry, but I would rather breed the farmer than the cow, because you can decide when the animal is bred the first time. You decide how long that voluntary waiting period is, right? That's actually a management decision. That's not the biology of the cow that's working there. Some people say that management of reproductive performance could make bad cows look good. So if you're using timed AI, or if you're having a shorter voluntary waiting period, there's some different management decisions that can actually affect what's coming out here. A long shot, some people say inbreeding. It's inbreeding's fault. Inbreeding depression is going to ruin us. And yet others say, and this is Rupert, by the way, Dr. Brookmeyer, who was here last year, I fought with him so many times over lunch. He said that breeding for longer teats, or shorter teats actually, has an effect on physiology. I'm not a physiologist. Sorry, I did it again. I'm not a physiologist. But I wanted to know, can we have data? Can we look at data that shows this? So let's start who thinks that number 1 is the most likely problem? Who thinks that it's number 2? Who thinks that it's number 3? Who thinks that it's number 4? Okay, so my ranking is pretty good here. All right, so let's look at short teats on physiology, the effects of breeding for shorter teats on physiology. We looked at this fantastic matrix that I showed you earlier. We can zone in on very specific relationships, and you can see here, geneticists love matrices, so you can see all the relationships at once. The relationship of measured teat length with all of these other reproductive traits that we have is pretty much non-existent. So the teat length thing is not a thing. Okay, good. Yeah, the reproductive physiologist says, of course it's not, you fool. That's fine. I'll take it. Next question. What is the effect of inbreeding on reproductive traits? Well, Baode did some work, Baode Macagnola. He did some work on looking at both production traits and reproduction traits. And then he looked at the relationship between pedigree inbreeding and inbreeding measured with genomic information. So a little bit more exact. And what he saw was indeed negative effects on the difference between the animals with very high inbreeding and the difference between those animals with high inbreeding and those animals with low values, inbreeding values. So there is a little bit of inbreeding depression, but the genetic gain from intense selection actually still far outweighs that inbreeding depression. There is no argument there. The monetary genetic gain for sure outweighs it, and these effects are real, but they are small. So, a little bit of the answer, maybe, but not a lot. Then we looked at timed AI. And timed AI is super difficult because we don't actually write it down. But what we could do is we can do simulations. And Dr. Gerson Oliveira here, he did a simulation on the effects of unrecorded timed AI. And he had a variation between the effects of the breeding values for calving to first service when no timed AI would be used, and what would happen if we had 100% of timed AI being used. And what he saw was that this difference here is actually, over 20 generations, the potential genetic gain that we lose by masking the animal's, I hate to use the word, but natural ability to get pregnant by using timed AI. Because that's really a management tool that you're using, but we can't separate out what's the genetic part and what's the management part if we don't record it. So this is kind of shocking, actually, because if we're not differentiating, I have nothing against timed AI, but if we're not writing it down, we can't differentiate between the animals that are really, truly, naturally super fertile and those that are not. Then Dr. Colin Lynch used some real data, and he looked at the effects of unrecorded timed AI, and you can see that the actual ranking of breeding values estimated using a data set that was recorded only using heat detection was not the same as the breeding values used under timed AI. So if this relationship here was a perfect line, we wouldn't have to worry about re-ranking bulls. But this is a snowstorm, right? You guys are in Michigan, you know what a snowstorm looks like. It's kind of not a line, right? And that's the problem that we see here. So overall, the results of all of this research is that data recording should probably include reproductive management to avoid re-ranking of our bulls and to have a better tool. So really, the true reproductive performance of our cows in this way is basically dependent on which timed AI protocol you might be using. And as a reproductive, I know Eduardo Ribeiro already told me that this is not a good representation of what a what a timed AI protocol looks like, but I thought it'll work. It'll work for all intents and purposes. So really, we're masking the true ability of an animal to get pregnant. So the summary, quickly, about these reproductive traits is that genetics won't help you, first of all, if you don't provide your animals with a good environment. That's clear. These traits are all very lowly heritable, meaning less than 10%. I can admit that. That means 90% of the variation is caused by the environment as well. Traits like calving to first service, conception rates, fertility disorders, those are all genetically linked, but only slightly. Inbreeding depression does negatively affect reproduction, but in a limited amount. Management practices do impact genetic evaluations if we don't account for them, and data recording is really important, basically. So we have an opportunity here with all of these challenges, and that opportunity is what can we do to improve our traits? What kind of traits would be closer to the biology of the animal? So we can come up with new traits. Now, the first example here is novel fertility traits, size and position score. You may recognize this rose here between two thorns. This is Augusto, one of your very own. He's between Ronaldo Seri and Audrey Martin, a former PhD student at the University of Guelph. And they work together to kind of move forward this idea of size and position score of the uterine tract. Okay, so this is an example of where veterinarians came up with a trait, and we geneticists could then say, oh, look at the potential here, right? We have different size and position scores of this uterine tract. You can see here, this is tightly between the pins, up quite high, and this uterine tract is quite deep within the animal, so you can just classify them according to one of those three sort of general areas. And what the veterinarians saw is that animals with size and position scores that are quite high up and smaller, they actually have a higher percentage of pregnancies per artificial insemination. Significantly higher than those with uterine tracts that are deep and further in the animal. So what's a geneticist to do? Well, we estimate heritabilities, right, Rob? That's the first thing that we do. So we did. And we can see that the amount of data here is low, but if we had repeated measurements per animal, we would actually be starting at a heritability of 10%, which is actually quite good. With a trait like that, it's a measurable trait on an animal. It doesn't cost a lot to measure, but we could increase the accuracy and the heritability of one of our fertility traits. If this relationship is actually true, then we could start saying, okay, instead of calving to first service, maybe we could start measuring traits like this that you could measure on an animal that actually have to do with whether or not she can become pregnant or not. Another example of a novel fertility trait is anogenital distance, which is the distance between the anus and the clitoris of an animal. There have been studies done previously to show that shorter anogenital distance has favorable correlations with positive reproductive outcomes. Again, a physical trait, a measurable morphological trait that you can measure, that you can quantify, and you can start to see how this could be incorporated into a breeding program. Looking at heritabilities, we can see that we have additive genetic variances here that are considerable. We can see that the heritability is considerable. And genomic estimates and genetic estimates, they are comparable. But this is also a trait that is closer to the biology of the animal. Far closer than anything that you could record just by when you're going to breed your cow. So a potential way to further improve fertility traits or reproductive traits. Again, here we have Augusto makes another appearance, looking at trying to incorporate sensor data into our reproductive performance breeding value estimation. We know that we can standardize phenotypes based on automated sensors. We can look at things like estrus expression and the relationship between estrus expression and embryo survival. Animals that show more intense heat tend to have more or higher embryo survival, which is fascinating. And the goal here would be to find genomic markers of estrus expression and fertility that can be used to select animals that hold pregnancy longer. We have developed a few different traits, like days between calving and first estrus activity, recorded with a sensor, not by a producer. We have maximum intensity of estrus behavior and duration in hours of estrus behavior, and we can estimate heritabilities. They're written here. And you can see, again, they're not perfect, but they're higher than the estimates that we have for things like days open, etc. So getting closer to the biology of the animal, letting the animal express her own fertility and measuring that is a better idea than measuring what the farmer's management decision was. Health traits. What can we do there? Any veterinarians here? You guys have the same problem that we do. We don't like each other, right? we don't deal with veterinarians at all, ever. Okay, so we need their help, right? I'll admit we need their help. We need to know what traits we need to measure. In Canada, we've done some work in this area. I'm sure in the United States it's very similar. But in 2014, we incorporated a sort of a sub-index called mastitis resistance. It involves clinical mastitis and somatic cell score. We have hoof health, metabolic disease resistance. You guys use milk fever. We use these different traits like displaced abomasum, BHB, subclinical ketosis, etc. Then in 2018, we incorporated the eight most prevalent hoof lesion traits into a hoof health index. Most recently, we incorporated fertility disorders like metritis, retained placenta, and cystic ovaries. And now we're working on calf respiratory disease, calf diarrhea, and leukosis. Who thinks this is baloney? Yeah? Total baloney. Fully, completely. I've got to be careful where I tread here. Now we've got a non-believer in the audience. Okay, so let's do it. Let's take this apart. Fertility disorders. Heritability, super low. Okay, 3% across all these different traits. Genetic correlations between first and later lactations. CO stands for cystic ovaries. MET stands for metritis. So there's some positive correlations between first and later lactations. Generally, cystic ovaries is not so correlated with metritis and retained placenta. But what we see when we look at the top, we can estimate these breeding values, we can do genetic evaluation, and then we can take the top 10 bulls and the bottom 10 bulls. So the extremes of our population. If there was no difference between the top 10% and the bottom 10%, then I would agree with you. But we see, when we look at percent of healthy lactations in the offspring of these bulls, we can indeed see differences. Meaning that, for example, I'm going to take the best example because I'm a bit of a cherry picker. When we look at cystic ovaries in later lactations, the percent of healthy lactations caused by the best 10% of our bulls, 95% of their daughters have a healthy lactation. And that is different than the 77% of daughters with healthy lactations coming from the bottom 10% of these bulls. I like it when I hear the, that's good. That's really good. Okay, so first example, right? Okay, fertility disorders. Then this is super cool, and this is still bloody. Okay, this is very new. This is very new research done by, he's already graduated, but the paper's not out yet. So this is Colin Lynch's work on the heritability of diarrhea and respiratory problems. He looked at dairy comp data of our Canadian herds, and he split up, because we don't record this in a standardized way. We don't record diarrhea in calves. We don't record respiratory disease in calves at the national level. But he looked at some data collected by Lactanet, and he had these recording thresholds. You had to have 1% of your herd had to be sick, or 5% of your herd had to be sick in order to even be included in this study. But what he found was that there was indeed heritabilities for these traits. So there is a genetic component to diarrhea and to respiratory disease. Now, the any epidemiologists here? Yeah, somebody wants to kind of half raise their hand. They're smiling back there. I'm not sure if that means they're telling jokes or if they're epidemiologists. Nobody's really sure. But the epidemiologists here will say, well, it depends on the disease pressure that the calves in a pen are experiencing, right? How on earth do you record that? Well, Colin tried to come up with a way to do that. And he looked at estimating heritabilities for susceptibility and infectivity for diarrhea and respiratory disease. Now, maybe the numbers are totally wrong, but when we talked to Andrea Dushel-Wilson from the Roslin Institute, she came up with similar numbers. So this is kind of cool, actually. You can model the effects of a pen. It's super difficult. I don't even want to get into the math here. Nobody really does, but Colin did for us, so that's good. Maybe Rob does. But what's really interesting here is that we see, these are real numbers. These are numbers that you could work with in a breeding program. 15% is quite good. So this is, of course, quite new, and we have to decide whether or not we want to be breeding for these types of traits, but it is an interesting approach to reducing the susceptibility or the infectivity, or both, for calf health. So the genetic aspects of health traits, genetics will not help you if you don't provide a good environment. Notice that's the second time that came up, right? These traits they have a genetic component. It's a small one. The environment plays a much larger role, but genetics can be a tool to help improve these traits. Then these traits also we in Canada use multiple different phenotypes to come together to have a subindex to breed for things like this. For example, clinical mastitis and somatic cell score will be used as a mastitis resistant subindex. It's a tiny little group. You shouldn't just use one trait and say this is going to be it. There are nuances to that. And again, data recording is truly essential for proper selection program. We can't get away from looking at a cow or a calf. The final piece of this presentation is now environmental sustainability traits. This is a story that loves to be told. The dairy industry has been very successful in terms of reducing the amount of resources going in to the system and maintaining or even increasing the amount of milk that comes out. This graph shows the number of dairy cows in Canada. The number of dairy cows is shown in the yellow line, and the blue bars show the total amount of milk in hectoliters. In 1960, we had about 3 million cows in Canada. Today, we have less than 1 million, and yet the amount of milk that these cows are producing has increased. What a story. What an incredible abundance of production from these animals. Fantastic. And yet, Google cow. Nuclear bombs coming out of their back end. If it's not the back end, by the way, you know this, right? The nutritionists will know this. This is not true. This is ridiculous, actually. A Taylor Swift concert will cause more climate change than our cows will. It's true, believe me. Actually, one liquid natural gas tanker that is in the ocean right now can't even hold, we couldn't even fill one of those liquid natural gas tankers with all of the methane produced by all of the cows in Canada. We couldn't fill one. And yet, this is the story. So, what do we do about this? Well, we have to work together. So we have this project. We had this project called the Resilient Dairy Genome Project. I'm going to use a very small project to tell you about our story in this, but the world has moved towards trying to breed for lower environmental impact from the dairy industry. And I'm happy to work with Mike and other researchers here from the United States. You can see that this is a pretty big circle of friends here who have worked together on this over many years. And I want to tell you about the Canadian side, the Canadian part of this. So be aware that this is indeed just a little tiny window into what's going on right now in the area of methane and climate change and environmental sustainability. And we're grateful for these fantastic partners because without them, we wouldn't have been able to do what we've done. And what we've done is we've made these relatively large databases on feed efficiency and methane emissions. These traits are super expensive. They're really difficult. The producers don't believe in them. They say this is stupid, frankly. They're kind of right, but we have to do this. We have to do this for social license. We have to do this because we can, and we can do better. So what this and a series of other projects has focused on is truly trying to make a big data set, basically. A big data set for feed efficiency and a big data set for methane emissions. Individual cows being measured across the world. And with that, we've done a little bit of research on calf efficiency, we've done rumination time, but most importantly, the industry in Canada has tried to develop a genetic evaluation system, as they have done here in the United States, for feed efficiency and for methane emissions, to limit the amount of environmental footprint that this industry is having. I cut all these slides out because I know that there's only a limited amount of time, and I could talk forever, believe me, but I won't. I won't do that to you. Ultimately, coming down to the methane emission evaluation, we have a feed efficiency evaluation. It's similar to the one in the States, but it's different. It was 2021, I think, that here in the United States you started breeding for feed efficiency. Same in Canada. We have a different approach, but basically you're trying to breed for animals that eat and use that feed to make milk. With regards to methane emissions, we use a single-step 4-trait animal model. We have predicted methane that we're using as the trait, so we're not even measuring methane on a per animal basis. We're using a reference population going through mid-infrared spectral data from milk samples and using that to select for animals that have low methane. Does it work? Yes, it does. People don't believe it, but we did a little test. This was recently presented at the European Association of Animal Science, where we took the SNP effects. So we estimated SNP effects in our population, and then we took it completely. All the Holsteins are related today, but we took animals from the UK, from France, and from Italy, and we took those SNP effects that we had estimated in our population, and we extrapolated them to those foreign animals to see if we ranked them according to our breeding value. Sorry, I did it again. If we rank those animals according to our breeding value estimation, would we be able to see differences in the methane production of those animals? And indeed, we have the groups of low EBV, so not low methane, but low estimated breeding value. So meaning these are not the good animals. They're producing 420 grams a day. And our high animals here are producing high breeding value. Don't be confused with the amount of methane. High breeding value are producing approximately 380 grams of methane per day. So even with animals that are not in our own data set, we can still use the breeding values to differentiate between animals that are high producers of methane and low producers of methane. That's in a nutshell. But basically, we're trying to predict methane production that's genetically independent of milk, fat, and protein. And the physiologists among you will know how difficult that is, because those physiological metabolic processes are tightly intertwined. Fat is not unrelated to methane production in animals. But this is how Canada has decided to go. The heritability of predicted methane is 0.23. That's the bus we're on, and it's driving. And we will continue to alter and adjust, but it's pretty cool, actually, that it works. So ultimately, the moral of this story is that now, when I Google cows and methane, I can see some new headlines. We're getting away from this nuclear explosion out of the back end of a cow, and we're getting a little bit more positive. And that gives me hope. The students this morning asked me what motivates me in my work. This type of change. You are able to make change. It's difficult, but you can read now, climate-friendly cows bred to belch less methane. That's a good story. That's a much better story than a green gas bomb out of the back end of a cow, right? I think. And what made this possible is teamwork, right? Researchers working together with industry, working together with farmers, all pulling on the same string in the same direction. So in summary here, again, data recording is essential for selection programs. You cannot move things if you're not collecting data. Both feed efficiency and methane have a genetic component. They can be incorporated into breeding programs. We have to work together. And again, data recording is truly essential for this. The best part of this whole story is that what we do in terms of research actually gets implemented into the real world. And your colleagues here, Rob and Mike, were recently awarded with a nice award showing the results of their implementation of their research into the real world. This has impact. In Canada, we're very proud to say that these efficiency traits, as well as the health traits, are being incorporated into how animals are being selected nationally. And that, to me, is far more valuable than any research publication that I could ever do. I won't get a Nobel Prize, nor will my colleagues that I've worked together on this, but I can take great pride in the fact that we've changed how animals get bred, and that will have an impact. And you all, with your work, will also have the opportunity to have this type of impact. Overall, genomic selection can boost these things. It's not the silver bullet, but it can be used to help move in the right direction. We need data-driven breeding for sustainable improvement. Accurate data recording does prevent bias in genomic selection programs, and genetic selection can improve how we breed our animals. With that, I would like to thank this absolutely fantastic force that is behind me. They have worked tirelessly over the years. This is not the current team. The current team is sprinkled in here, but these are graduate students, postdoctoral researchers, research technicians, and fantastic people who have actually done all the work that's allowed me to stand here in front of you today and show off. It's because of them that this has happened. And of course, our funding and organizations and collaborators who also have contributed immense amounts of discussions and thoughts and data to this work. And with that, I would like to thank you so much again for the opportunity and your time.